Yeah, let me know when you're ready.
Okay, we are now recording, and thank you very much, Sarah, for giving a Build a Soul seminar, and it's all yours.
Excellent. Thanks, Kate, for having me. And thanks, everyone, for being here today. So I am a theoretical physicist by training, but I primarily work in astrobiology. And so I'm an associate professor at ASU. And I mostly focus actually on origins of life and trying to understand if there are universal principles that underlie living systems. So sort of the core focus of my research is actually looking for whether there are mathematical laws that describe biological systems that would apply not just here on Earth, but anywhere in the universe, and also help inform how we can build quantitative frameworks for understanding the transition from non-life to life. And so that's led me to a lot of interest in just understanding fundamental properties of biochemistry, kind of more from the perspective that a physicist would take about thinking about regularities or law-like behavior. And that's mostly what I'm going to talk about today, although my group does a lot of other things in other areas. But I thought this was going to be the most interest to this community, as far as thinking about how some of these sort of more abstract ideas about the nature and organization of life might actually lead to new principles that could be of use in design of synthetic cells. And so just to start off, I always have do a little bit of philosophy at the beginning of my talk, but I've kind of structured it to be a little less philosophical than usual because I know this community is a bit more on the practical and applied side. But I do think it's kind of important to ask the question, what is life, when we're talking about building life, and to really be sort of concrete about what the problems are with understanding what the phenomena we call life is and how we should be thinking about it from a first principles perspective. So this question is obviously a very old question, and just from the perspective that I ask, which is kind of looking for these ideas of laws or universal behavior that might lead to an understanding of life. I think Erwin Schrödinger's book that was written in 1944 after he delivered a series of lectures on the topic, What is Life, in 1943 is a good kind of conceptual place to start, at least as far as how the modern question of what is life has been asked from this kind of more theoretical or abstract perspective. And the question he was really focused on is how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? And that book was actually incredibly influential for among many reasons. He was able to sort of logically deduce, based on the constraints of the laws of physics, what the structure of DNA should be as a aperiodic crystal. And that inspired Watson and Crick to look for the structure of DNA, which they discovered along with Rosalind Franklin. So that was a really influential aspect of this book, but he also introduced this concept of negentropy. But I think from my perspective, some of the things in the book really didn't go far enough, and they really aren't maybe the best frameworks for thinking about the kind of questions that we're really confronted with, especially nowadays, in understanding what life is and really trying to build original life experiments or build life in the lab, and also to look for life on other worlds. And one of the reasons is for this very what I've highlighted here, that he was really focused about within the spatial boundary of a living organism. And I'm not sure that's really the right scale for thinking about the problem. Of course, he's not the only person to have thought fundamentally about what life is. There's a lot of attempts to define life. This is just one example from a short paper by Carl Sagan on definitions for life, which is a nice little read, and I really recommend it. It's only 2 pages, and it's a little cheeky, so it's kind of funny. But this definition of life that he provided is kind of one that you would read in a biology textbook, right? So it's any system capable of performing a number of such functions as eating, metabolizing, excreting, breathing, moving, growing, reproducing, and being responsive to external stimuli. And it seems pretty straightforward to list all these properties of life, but of course, if you list those properties, you might end up with paradoxical examples. So the one that Carl Sagan cited in that paper was that based on this definition, if aliens visited Earth, they might think that cars are the dominant life form. And in fact, now cars have even been to space. So perhaps, they might be even more confused if that were their definition for life. So the really sort of hard problem about thinking about a lot of the issues that we have when we're talking about origins or synthetic life or looking for life is that we don't have a viable working definition of what the phenomena is that we're interested in studying. And I think that in that essay, Carl made a really important point about why definitions for life seem to be inadequate, or in fact fail in their attempt to actually draw boundaries around this phenomena that we call life. And so the point that he makes is that many such properties, like the ones he listed in the previous definition, are either present in machines that nobody is willing to call alive or absent from organisms that everybody is willing to call alive. So I actually disagree on the point about machines for reasons that I might make clear in the next few minutes. I actually think machines should be included in the category of natural phenomena that we call life. Um, uh, but I think the point that he was making with this definition is actually runs a little bit deeper about discrepancies about sort of categorization of specific things that we may or may not think need to be included in the end. And basically the point he was trying to get at is if you start from this kind of axiomatic approach where you're like, these are the things that I want to include in my definition of life, and you start with some kind of descriptions of those things, you end up with a description of life that includes cases that you meant to exclude and excludes cases you meant to include. So it seems to be that it's really hard to draw these boundaries in a hard way, even if you start from reasonable starting points and trying to build your definition. And this is one of the reasons that viruses have been so hard, because they're in this kind of gray area where they don't fit these concrete hard boundary definitions. And from my perspective, I think part of the problem is that we've been attacking trying to define life rather than ask questions about what are the fundamental principles about how the universe works that we might be missing from which we could derive properties of life. And so this is very much the perspective of theoretical physics, that there are phenomena that we don't understand in the world, and we should be able to derive their properties from some explanatory theory. And so one example I often give with this is just to sort of think about some of the history of development of thought in physics. A good example is studying planetary motion, which 2000 years ago people thought was governed by perfect circles, because perfect circles were, perfect like the heavens, and so state-of-the-art predictive theories for planetary motions were to draw circles inside circles, which we call epicycles, and in ancient times. And that was sort of the dominant model for planetary motions for almost 1500 years or more than 1500 years actually, till Galileo and Newton came along and thought about terrestrial and celestial motion and how they should be unified. And then Newton came up with the laws of motion, which are universal, and in particular laws of gravitation. And then you had further abstraction of the ideas in the 1900s when Einstein came along and realized, studying properties of a very different phenomena, properties of light, that space-time is actually curved. And that really explains planetary motions, but also me sitting in this chair. My reason for this sort of aside is that I think these kind of definitions that we've been talking about are kind of at the epicycle phase of development of intellectual thought. Like we have really good descriptions, and sometimes they can be predictive, but they're not explanatory. And so what I'm interested in is trying to dig a little bit deeper and figure out what is the explanation for life, and is it some phenomena that it might be something that's not readily perceptible to our senses, in the same way that curvature of space-time is not exactly my experience of being stuck to my chair, but that is a really good explanation for why I'm sitting in my chair right now, and very predictive and explanatory of a lot of phenomena, and allows us to extend further into examples of things that we haven't observed yet. And that's really the power of physics. And this kind of way of abstract thinking is it allows you to take examples you have, figure out principles or explanations for those that allow you to extend to examples you haven't seen yet. And so that's kind of my interest, at least coming in this from a theoretical physics perspective. But other people have also kind of taken the viewpoint that definitions for life are really not helpful. And these are just a couple of my favorite quotes on that. One of them is from the American Chemical Society meeting in 2012, where Andrew Ellington was giving a talk on, some of the stuff he does in RNA chemistry related to origins of life, and made the radical claim that life does not exist, which I think he still holds as a view, and I'm maybe coming around to agree with, at least not in the way that we think it does. And I think this was actually really nicely captured in this short essay by Jack Shostak about why we should not worry about defining life in order to study its origins, which I also originally disagreed with, but now I'm kind of coming around to understanding really the key points. Where he says, as one focuses experimentally on any of the quote unquote defining properties of life, the sharp boundary seems to blur, splitting into finer and finer subdivisions. So the point here is really just that life is not a property of molecules. It's what we would call in science sometimes an emergent property. It's a system level property of collections of molecules and reactions. And we haven't really figured out a way to systematize or regularize an understanding of what life is as that sort of a property that emerges in chemistry. And there's been a lot of focus in definitions of life as a chemical phenomena. But I don't actually think that life is chemistry. I think life is actually a representation of physics of information, which I'll discuss in just a minute. And that physics, actually, the first scale of physical reality where that physics actually emerges is in chemistry, because that's the first place where you have enough combinatorial diversity for information to actually matter in some way about specifying the outcomes of specific processes. So really sort of the idea I'm interested in is sort of like why I think this might be useful to this community to think from the sort of more abstract perspective and then try to bring these things in, is that when we're talking about the design principles for synthetic life, right now we're really focused on like, what are the biological parts that we can put into a system? So I always love the images that Kate uses for your Semini series. They're phenomenal. So I stole the one that she used for this talk on Twitter for this. But it's not just enough to have the parts. We have to have the principles about how life works and what life is. And so just as sort of like one component of that, I wrote this essay a few years ago, which is a little bit abstract, but I think gets at some of the key points that we should be thinking about. Is when we're talking about life, we're really talking about different sorts of things that might come together to build an explanatory theory, but we kind of think about them as the same concept, and disentangling some of the features of life might be really important. So Michael and I tried to make this argument that we really should sort of talk about two separate concepts when talking about trying to build theories for life, and we called them life and alive. I'm not beholden to those names, but I think it's useful to kind of provoke new thinking about the problem. And part of the argument was that life is the sort of set of things that require information to produce them in the universe. So a synthetic cell right now at sort of level we can build it with biological parts is certainly representative of life from that perspective, but so is my cell phone, because my cell phone is a product of an evolutionary process and a long acquisition of information in the physical universe to be able to construct a phone. The example I have here on the right is a little bit more abstract, but related also to this idea that information is necessary for certain processes to exist in the universe, or certain physical structures. So the argument that I make here is that anti-accretion, which is something our planet is doing by launching little metal boxes into space, which we call, the space program and launching satellites, requires information in the form of knowledge of the laws of gravitation. It's a statistical regularity that we've observed in the physical world, that physical systems have extracted through evolutionary history and through learning, and now allow this process to happen on our planet that wouldn't otherwise. I would argue that making a protein is also a similar sort of process. You don't expect a very large macromolecule to spontaneously fluctuate out of a chemical soup. It's so statistically improbable to have that molecule form in high abundance that the argument that we're making is that it's actually not probable unless you have a system that has acquired information about how to build that specific object. And so this is sort of part of the way that I'm trying to tackle thinking about how to construct a theory of life. And so the distinction with alive here is then living things, or things that are alive, or things like you and me that are actively information processing, actively constructing ourselves. So I can think about myself as an informational imprint on matter. I don't have the same atoms in my body that I had 10 years ago, so I can't think about myself as the same physical stuff. But there is a continuity across space and time that is a thing that we call Sara, which is the fact that I am a system that has these kind of informational patterns that keep rebuilding myself. So I'm life, but I'm also alive. My phone might be life, but not alive. A synthetic cell right now might be life because it's a system, again, that's a product of evolution. We took all the parts from this long evolutionary history of acquisition of information, but what we want to do is try to figure out how to make those alive. How do they become self-sustaining systems that can imprint their own information into their physical structure? And so I very much take the view that there should be fundamental principles that describe these sort of abstract ideas and actually make them concrete and testable. And so this comes from a long tradition of physicists that think, there should be general laws that are mathematical in nature that can describe all the phenomena in the universe, including life. And so I rather like this quote from Albert Einstein, where he really does try to think about including it in terms of how we think about the structure of fundamental physics. Um, and in physics, we actually have this kind of idea that comes from John Wheeler about it from bit, that the entire universe should be sort of information theoretic in origin. And so part of this proposal is there is a fundamental physics of information. We don't understand it yet. If we understood it, the kind of structures we would study in the physical world that are representative of that physics are living things. So it's a little like we are accepting of the laws of gravitation as being universal, and we study massive bodies like planets and black holes or galactic scale phenomena to study gravity, but we agree that gravity doesn't, it's not important in the nucleus of a atom, right? So gravity is a very long range force, requires very massive objects in order to become a dominant force that we use to describe those kind of systems. The proposal here is sort of when you get into chemistry and you get high combinatorial diversity, it's the possibility space in chemistry for, I'm sure many of you know this, is exponentially huge. So there's estimated like 10 to the 60 molecules under molecular weight 500 or something. So so it's actually bigger in some sense than physical space. If you could compare geometrical space to a combinatorial space, I think chemical space is much, much bigger. And so part of it, and then if you get to the objects like cups and people and cell phones, the combinatorial space is actually even larger. And so part of the argument is when you get the transition to the origins of life, like the transition we call the origins of life, what that transition is from a more fundamental perspective is where information has to become the dominant physics, because those things would not exist without some imprint and other physical structures for them to exist. And so there are ways about talking about these abstract ideas more rigorously from an informational perspective. And we're working on this with collaborators, actually with Kate too, and another collaborator at University of Glasgow, Lee Cronin, that the three of us have a project together to try to think about quantifying the transition from non-living systems to something at least as simple as a minimal synthetic cell from this kind of informational perspective, using something that we call assembly theory, which I'm not going to talk about in this talk because I want to talk about things that are a little bit more concrete from the biochemical network side. And I'll get to that in a minute. But in any case, I just wanted to just bring this up as sort of an introduction to my talk, because I think intuitively we think we know what life is because we are life and we experience so much life in our day-to-day experience. But I don't think that we should make so many assumptions about what the key properties of life are, but that we should try to be able to derive them from a more explanatory framework. So again, that's my bias as a theoretical physicist. Of course, this research program might not pan out, but I think that this is a constructive way of thinking about it. And that, in fact, rather than asking what is life, someday we might ask life is what, because it might be a very counterintuitive explanation. And so I like this quote that was at the end of Schrodinger's book, where he appeals to possibly of other laws of physics necessary to explain life. And that's actually what I spend most of my time thinking about. And so then the proposal becomes the origin of life is finding the transition from non-living to living physics. So you have abiotic systems that don't have information being really necessary for those objects to exist in the universe, those molecules. They can form readily by stochastic random chemical processes. But when you get to a certain threshold in this space of possible molecules, it gets too diffuse in the space. There's too many combinations and too many possibilities. So if you had a random walk through that space, it's just the probability would be so low of forming anything, let alone in high abundance. And so the question is, what is it that transitions those systems across that boundary? And that's really sort of the key aim of the physics that I'd like to try to understand. And like I said, we have a lot of ideas about what that physics might look like and are building theories actively that are coupled to experiments. But what I'm really going to talk about today is sort of, so these are sort of the 4 areas of research that my group focuses on, which are all tying into this sort of overarching conceptual framework that life is really the physics of information and trying to pursue it from different perspectives. But what I want to talk about today, because I think it's sort of the most concrete and most useful thing for this community, is really this idea of statistical approaches to characterizing life's chemistry. And so what I mean by that is really trying to think about not the specific biochemical components in life as we know it, but can we abstract any statistical regularities that tell us general principles about how biochemical systems should be structured, even if they have very different chemistry. And so I think about this in terms of a concept in physics, which I'll introduce in a minute, which is called universality. But of course, we do have notions of universality already in biochemistry. And a lot of these come from the fact that we have a last universal common ancestor. So shown on the right here is just an artistic representation of the tree of life. And we know if we trace back in time all the genomic information in modern organisms, we can recover properties of what's called a last universal common ancestor, which is probably a community of cells or cellular like structures, or maybe not even cells yet, or individuals in the sense that we know now, that had a lot of the modern biochemical machinery. So for example, used DNA and RNA and proteins and, had ribosomes and all of those features that we associate with being universal across life on Earth. So this sort of informs the model of universality in biochemistry, or universal biochemistry, as Norman Pace has called it. And he makes the argument, it seems likely that the basic building blocks of life anywhere will be similar to our own in generality, if not in detail. And so here, we know life on Earth uses certain biochemical components. And we know that forms sort of the universal model of biochemistry, at least for life on Earth, but we don't know how to abstract that to what alien biochemistries might look like, or what the chemistry of life was at the origin and what its properties were. Or if we want to design synthetic systems, how do we move beyond those components? Or how do we know we have the right set of components to actually make a living system and not just have it be a product of life and actually have its own sort of autonomy and living properties. So this kind of way of abstracting from specific examples and thinking about new ones, I think will really come from, as I just said in a minute, and I'll kind of explain as we're going through, thinking more statistically about what biochemistry is. So I mentioned before that we usually think about life as an emergent property. It's a systems level property. And in some ways, life doesn't depend on the specific molecules or reactions that it's involved with. Like right now, the model we have does depend on those features, but you could imagine, or at least hypothesize, it could be the case that there's actually patterns in the chemistry that are the things that are really indicative of having a living system. And those patterns could be moved to other chemistries and also display what we would call living behavior. So part of the inspiration for this kind of line of thinking comes from the way we think about universality in physics, which is quite different than that model of universality from biochemistry, which is focused on the same components being repeated across many systems. So that's sort of universality and component membership. Every living thing on Earth has the same component parts, essentially. In physics, we talk about universality classes when the details don't matter, but there's some macro scale or statistical level feature of systems that is universal across different systems. And so just two examples here. This usually appears in sort of the physics of phase transitions, where we talk about this notion of universality classes. But you could have two very different kinds of physical systems, like a liquid gas critical point, so the phase transition between liquid and gas, or the ferromagnetic critical point, which is when you have a spin up, spin down system, and you're talking about it going from the ordered to the disordered phase. If you look at the behavior of certain features of those systems as a function of temperature, they have the same critical behavior at that phase transition with the same exponent. And so they're said to be members of the same universality class. And this is really an important concept in physics because it allows us to unify properties of very disparate systems within sort of the same structure. Mathematical structure at this sort of statistical level. And then it also allows us to predict properties of new materials we haven't even observed. And that's the feature that I think is really important, because once you're not talking about the details of a system and you're talking about these macroscopic or statistical regularities, it allows you to talk about what systems are consistent with those kind of patterns. And could those be living too, if we think about a universality class for biochemistry, a universality class for the chemistry of life. And so that's sort of the question that my group has been asking a lot about lately, is trying to think about statistical approaches to characterizing biochemistry. And we might cover some of the features that we already know about biochemistry in terms of universality and things, but it's also a way to try to extrapolate what are the principles underlying these systems at this kind of, in terms of statistical regularities that we can allow to apply to other systems. So to ask this question, we have to sort of acknowledge the fact that life is at least at the fundamental level of description, like where life emerges in physical reality, it's in chemical space, right? And so I can't really talk about the space of phones and cups and things as easily as I can talk about the space of all possible molecules and their properties. And so I really like thinking about this notion of chemical space that comes primarily, I think, from the field of pharmaceutical drug design, or at least that's where I read most of the papers about chemical space. And defining its properties, but in particular to think about the biochemical space is defined by life on Earth. So we know of a lot of examples of biochemical systems on Earth now, and they basically define a set of molecules that life uses in this chemical space. And if you think about chemical space as an actual space in a mathematical sense, you could talk about what is the shape of biochemistry in this space, at least biochemistry as we know it. And is it possible to build other systems that have that same shape in chemical space, but have different component parts? And so that's sort of the way that I'm thinking about this. And so to represent some of the features of the shape of biochemical space, my group works a lot with networks. So this image I'm showing here is essentially all of the data in the KEGG database, the Kyoto Encyclopedia of Genes and Genomes. And so that database actually catalogs enzymatically catalyzed reactions that biochemistry uses. So we're dealing mostly with small molecule biochemistry here. You can think about it as a metabolic network, if you like. This is a planetary scale metabolic network in the sense that it has no notion of individual or community here. It's just generating all of the things that biochemistry can catalyze and generating one large network structure to represent that. And the representation is actually such that each dot here is a molecule that biochemistry uses. They are connected to each other if they participate in a common reaction. So we call that an edge in the graphical representation. And the size of individual circles here are what we call nodes in the graph, correspond to how many reactions that molecule participates in. So some of these very large ones are what are called hubs in network theory, and they're things like ATP and water because they participate in a lot of biochemical reactions. So this is a way of sort of mathematically representing the statistics of the patterns of interactions that molecules have in biochemistry. And there are some other features that are more related to the specific kinds of molecules or reactions that biochemistry is using that I'll talk about in a little bit. But the first part of the talk is really just going to be talking about whether or not there are features of how life has these patterns of interactions in biochemistry that we might think of as universal organizing principles for biochemical systems. And so the way that we do that is to think about, so that was a planetary scale network that I showed on the last slide, but you can also think about biochemical networks at the scale of individual taxa or species, and also at the level of communities. And so we get data from the Joint Genome Institute database that catalogs genomic data, and the first results I'm going to show actually have the statistics presented here, but some of the later ones we have more data because JGI is being updated all the time. So sort of the most recent data sets we have actually have like 11,000 metagenomes. And for those that don't know, so the individual species have genomic data in this database, and then metagenomes are just community samples that maybe are taken from an environment like a hot spring, and you don't have any knowledge of the individuals, at least when you do that sampling. And so you can talk about sort of the community level genome. And then the biosphere, as I mentioned, is all of the catalog enzymatically catalyzed reactions. And so there's a few reasons for looking at biochemical networks across different scales. One is that what I'm going to present is actually scaling laws for biochemistry, which we are using to try to derive theory that could predict the properties of the scaling laws. And for those that don't know what scaling laws are, I'll just explain that in just a minute. But there's also this kind of idea that comes also from physics, that if you see scale invariance in a system that has the same properties independent of the scale you look at it, it might be more likely to be universal. And so we do have this issue in biochemistry of getting around the universal ancestry of life on Earth to try to talk about properties that might apply to other biochemistries. And so I think this idea that life might have organizing principles across different scales means that those features might be much less evolutionary contingent than, say, if you just looked at the properties of individual taxa or species, which we know are subject to selective constraints, and will follow that sort of lineage that we saw before. So there's constraints that operate across different scales in biochemistry. And certainly the biosphere is not evolving in a Darwinian sense in the same way that individual species are. So if you see constraints across those different scales, it suggests different constraints on sort of universally operating on evolutionary processes. And so, so how we're thinking about looking at this ensemble across scales and thinking about biochemistry exists sort of in the global network of all catalyzed reactions that biochemistry uses is this idea of planetary systems biochemistry and looking for scaling behavior across the different levels of organization in the biosphere. And so here I've just written a network property, like it could be how connected is the molecule, like how many reactions does it participate in, and then average that over, say, the whole biochemical system of interest, whether it's an individual organism or a community. And then you look at it as a function of the network size, which would just be the number of chemical species that participate in that biochemical system. You think about it as the number of compounds in the metabolism. And we do that for individuals, ecosystems, and then we also look at how the biosphere sits on those scaling laws. And sort of the hypothesis we had originally was if we see universal behavior across all of these scales and it's different from random chemistry, then those would be the things that would be good candidates for universal organizing principles or universal laws of biochemistry. They would be things that we might expect to apply to other biochemical systems, or at least if we could derive theory that could explain these particular scaling behaviors, we would be able to generalize that theory to apply to different biochemical examples, because there might be some variation in the exact scaling coefficients and things that you could derive from a particular theory. So this is sort of the idea behind scaling laws in biochemistry. And just as an aside, scaling theory has been really successful in describing a lot of features of biological physiology and is sometimes described as sort of the only law-like property that biology has. But just for example, like body mass and metabolic rate scale in a very predictable way. And there's some theories that have been proposed to try to predict why that's the case. And even like global microbial diversity has been shown to scale and possibly predict missing diversity in the biosphere. And we don't always have to just do network properties here. They could just be things like number of reactions or other features of the system. And the network size doesn't have to be the only thing that you're looking at as far as the scaling parameter. It's just that you want to look systematically at the systems and say, if I vary this parameter, does this parameter vary in a very regular way? And there's no reason to assume that's the case. So just as an example, in biochemistry, we know the genetic code doesn't scale. It's kind of a fixed property of all different systems. So it doesn't observe regular scaling behavior. And there's lots of other examples that you can think of in biology where there isn't scaling behavior. So it's not a trivial feature of a system. It really is saying something about organizing principles. And so what we see in terms of the biochemical ensemble that I presented before is that there is universal scaling and network topology across individuals and ecosystems. So these are just two network measures. This is average shortest path length. So if I look at the network and I say I take the shortest distance in terms of connecting two molecules by a set of reactions, that would be the average shortest path length over the whole network. And then clustering coefficient basically measures how many molecules have a neighbor shared in common. And we looked at a whole bunch of other different measures, like I said, reaction diversity and other things that don't even necessarily have something to do with the network properties themselves. And what we see is a lot of the features do scale in a very regular way, and you can assign power law scaling behavior to the overall global topology of these networks. So the networks are actually changing in a systematic way as you change the size of the network in terms of the average properties of the statistics of the patterns of molecules in terms of their interactions. So that kind of confirmed the idea that there could be universal properties that constrain all biochemical networks. And a lot of these features actually might arise because we know that individual organisms or even communities are partitions of this sort of global biochemical space. So each living system is using some subset of reactions, but you'll probably notice by the sort of animation of all these organisms, like where they exist in biochemical space, they share a common subset. So this is the idea of universal biochemistry being brought back in here. So one question we could ask is this scaling that we see that's very regular across all these systems arising because of this universal subset of shared reactions, or is it instead a product of some other constraints on the system? And we can also ask, is it something that we would expect to characterize random chemical systems? Because if it's random chemical systems don't share the exact scaling laws, or they don't scale at all in a regular way, it would suggest that these are principles that apply just to evolve biochemical systems. And so what we did was actually randomly sample biochemical space. So there are a lot of chemical databases out there like Reaxys and PubMed, or PubChem, sorry. And the NIST database that have, large fractions of catalog chemical space available, but it's very hard to construct control samplings for these kind of processes. So what we did and we're using those for more recent work, but what we did for this particular paper was just sample biochemical space, but do it randomly. So we took random sets of reactions that were the same size as the real biochemical systems and just asked, do they scale the same way as biochemistry does? And what we see is that they scale, but they don't have the same exact scaling coefficients, and sometimes they're not having the same scaling behavior as far as being superlinear, linear, or sublinear. And so we can basically conclude that the features of biochemical scaling are not features of random networks, even when you constrain yourself to biochemical space as we know it. If we broadened it to larger chemical space, I suspect you would find even really different behavior. So shown on the right here in orange is the random networks, and the gray is just the ensemble of the original biochemical network data. So we can also do something where we can control for the fact that some reactions are found more frequently across life on Earth. And so this would be, we're not sampling actual organisms or communities like we did with genomes and metagenomes, but if we just sample random sets of reactions where we choose reactions to be more frequent in members of that ensemble, if the reactions are more frequent in life on Earth, then we recover most of the features of the scaling behavior, but it's not exactly reproduced. So there's some variation there. You still can kind of statistically tell the difference between these random ensembles and the real ones, but most of the features are recovered. And in fact, I don't show it here, but at the level of looking at network topology, you can also statistically tell the difference without knowing anything about the molecules or reactions participating, just the network structure between archaea, bacteria, and eukaryote. There's like a statistical confidence you could say that a network was drawn from one of those domains. So these are real features of the organization of biochemistry that have are manifest in sort of evolutionary differences between these systems. So just to sort of summarize that, what we have is these random sampling reactions are pretty far from the scaling behavior, and it's even farther still for some of the results I'm not showing. If we sample by frequency, so we're kind of trying to conserve that universal core of biochemistry across the data set in the way that it's conserved across real biochemistry, we get more of the features. And then a final sampling we did was to randomly sample genomes to see if the scaling was like real metagenomes or metagenomic data. And it recovers most of the features again, but you still can, at a statistical level, if you look at the properties of the ensemble, tell the difference between real ecosystems and these contrived ones. So it is telling us something about sort of the constraints on biochemistry and some of those features. The feature I think I find most interesting is that the scale-invariant topology, so these scaling laws that we see, a biochemical network structure across individuals and communities all the way up to the planetary scale, requires a universal set of biochemical reactions across life on Earth. And so one of the reasons I think this is interesting is because we can think about the universal common ancestor, not from sort of the evolutionary bifurcation perspective, where we had sort of a community of cells from which everything else radiated in terms of, evolved from ancestor, which is certainly true, but this adds a little bit of a different perspective to the story about why this universal set exists, is that perhaps the last universal common ancestor emerged in part because you need this scale invariant property of biochemical networks. And there's a lot of reasons for hypothesizing that, associated with the fact that the topology of biochemical networks is one that happens to be more robust to perturbations and things, which comes from a lot of work in network theory. So you can think, if you wanted to have biochemical systems that were robust across scales, for example, that they would have to have this kind of same structure across different scales. And that enforces the lower level units to have a universal set of shared reactions. So the argument I'm proposing here is that there are constraints at the level of communities and even planetary scale biochemistry that enforce lower level units to have some shared components. And there are some tests accessible things there that you could presumably do with ecosystems of synthetic cells in the lab, which Kate and I have talked about a little bit. And so I think this actually has real consequences and things that we can test as far as developing ideas about why the last universal common ancestor emerged from these kind of random chemical networks. And this idea of a phase transition really comes more from work by Goldenfeld and Woese on the idea that the last universal common ancestor was a phase transition to Darwinian evolution where individuals emerged. And I think this is consistent with it. It's just trying to show from the structure of the actual biochemical networks that themselves what some of the properties that phase transition might be. Okay, I'm going to go a little faster. I've probably been going fast, but I don't want to run out of time and make sure that we have a little bit of time for questions. So I will just mention briefly that one of the features that leads to universality is the fact that we can coarse grain things. So this again goes to the idea of looking at statistical patterns. And you can think about that in statistical physics just in terms of the fact that temperature is a really important predictive variable in statistical physics and thermodynamics, but it's not a property of the microstate, it's a property of the macrostate, right? We can measure temperature in terms of the average kinetic motion of particles in a box, which means many boxes that have very different configurations of molecules can have the same temperature. And so what we're trying to do is find out what are those coarse grain variables where biochemistry could be in these boxes instead of particles in a box, and they would all have sort of the same average properties, but maybe different chemistry. And so one of the ways of thinking about coarse graining chemistry, biochemistry, actually comes just from the way that enzymes are already cataloged, or at least enzyme functions are cataloged in a lot of these databases. So if you go into the KEGG database, every reaction is cataloged by an EC number, which is the enzyme commission number that's associated to the function of that reaction. And those functions are actually cataloged hierarchically. So you have a specific 4-digit number that specifies the specific properties of the reaction. But at every sort of scale above that, where you lose a digit in the EC number, you lose specificity about which specific reaction it is. Until you get all the way up to the level of classes where you have a very broad categorization of reaction types that biochemistry uses that are all bin together under this category. And so you can think about this as sort of like a coarse graining that we think about in physics because it bins reaction types together by their function. And there's 6 different EC functions that we've been looking at. There's actually a 7th that's now cataloged, but there wasn't enough actual data in the genomes and metagenomes of this function for us to use it. But these are the 6 that we were looking at. And what we see or basically the procedure is we want to coarse grain biochemical space in terms of functions now, instead of looking at the reaction topology. So that's one way of coarse graining biochemical space. This is another. We basically count each enzyme type in a genome or metagenome, and then we use that to basically build scaling laws for the number of functions in a particular class versus the total number of functions in that biochemical system. And what we see here is that we also get really regular scaling behavior across the 6 classes. And I can actually show the scaling coefficients that we get. So they're all power law behavior. We've tested other fits, but power law was the best fit. And what we see is that these scaling laws do reveal the existence of biochemical universality classes. So where we see coarse grain variables in terms of enzyme functions, or the properties of the reactions themselves in these networks displaying regular scaling behavior. And the notable examples that have universal behavior as far as being superlinear, so growing superlinearly as a function of size of the system, versus linear versus sublinear, are the oxyreductases and the hydrolases always scale superlinearly across all the data sets. So we think that's a sort of universal behavior of those enzyme functions, that they need to scale superlinearly with the size of the system in order for a biochemical system to function. And the ligases and transferases are always sublinear. And then with the lysase and isomerases, we get this kind of behavior where eukaryotes act a little bit differently. And then for the lyases, also the metagenomes act a little bit differently. The isomerases actually might also be universal behavior because this is actually consistent with it being sublinear for this category. So basically the different biochemical networks display these behaviors that we see universally across the data sets, which suggests that they could be features indicative of universal constraints on biochemical architecture. So you could think about evolutionary systems evolving towards some optimized limit, that the scaling behavior is actually telling you what that optimized limit is, because it's sort of a hard boundary on the behavior of biochemical systems. And so that's actually a hypothesis that also could be tested in the kind of systems that you all work with. But I just want to also highlight that why we think that this coarse grain feature is actually a universality class like we have in physics, and not one like we have in just because we have universal shared components in biochemistry. And the way we got at that was to actually look at enzyme universality across our data set, which is to say we counted the number of function, or well, so for each function, biochemical function in each enzyme class, we rank ordered it depending on whether it was present in 100% of archaea, for example. So I'm not sure if you can see my cursor, but where the archaea, so the data point of 1 on the x-axis of any of these plots means that that enzyme function is found in 100% of that data set. And then we basically rank ordered enzyme functions by how universally distributed they were across each data set. And then once you do that, you can assign an area under the curve score, which basically tells you how universally distributed functions within that enzyme class are across that data set. So if the AUC score is 1, it means that every enzyme function is found in every member of that data set. So if, for example, we had, the oxidoreductases had a AUC score of 1 in metagenomes, it would mean that every redox reaction was found in every metagenome. We don't see anything that's universally distributed in that way. And if it's closer to 0, it means that the component reactions, the functions, are less universally distributed. And there's a lot of information and sort of rich data in the things that we've been looking at, but just to sort of highlight the main trends, what we see is that metagenomes always have more universal components than individuals. Maybe that's expected because they're composite of individuals, but it's also an interesting feature about metagenomes being sort of more regular across different systems than individuals are. And also that the ligases, the oxidoreductases, which are in purple here, tend to be much less universally distributed than something like the ligases, which is also sort of intuitively consistent with what we would think, because ligases are known to be conserved in terms of their function across many systems, which is one reason that they're very prominent in constructions of the last universal common ancestor. And redox reactions tend to be more specific to the kind of metabolism that an organism uses. So you wouldn't expect as many shared reactions across taxa or even across metagenomes, because they're very environmentally dependent. So these are sort of consistent with what you might think intuitively, but the important point here is that these features don't track to the scaling laws at all. So for example, the oxidoreductases have the tightest scaling behavior in their exponents, and what we see sort of across the different data sets, but they're the least universally shared class. And ligases actually vary, even though they have universal scaling behavior, their scaling coefficient varies by 0.25 across the different data sets, and they are very universally shared. So there's not a direct correlation between universality of component reactions and universality of scaling behavior. They're separate properties of the system, which is suggestive to us that these scaling laws we're seeing actually are representing constraints on biochemical architecture and are an artifact of the fact that we have universal biochemistry. And we can look at that a little bit further by talking about current LUCA models. So we have some work that we're doing with Aaron Goldman, who's worked a lot on reconstructions of the last universal common ancestor. And he has a consensus model that is basically takes the 8 current LUCA models, groups them together, and then finds the sort of functions that are common across them and builds a consensus model. That data is shown in the star here as far as how that it would fit on the scaling curves. And what we see is that the last universal common ancestor, at least our current models for it, are consistent with the scaling behavior across these data sets, with the exception of transferases and ligases. And the ligases are maybe a little bit easier to understand because, again, I said that they were universally more conserved across taxa, and therefore they're more likely to be represented in the last universal common ancestor. And so one perspective you could take from this, which is one of the things that I think is interesting, is could these scaling behaviors constrain models for LUCA and allow us to develop new models for LUCA that satisfy these sort of universal constraints, or even extrapolate beyond LUCA to earlier biochemical systems. Maybe the ligases are overrepresented because of phylogenetic history and the way we reconstruct the last universal common ancestor right now, but the scaling law behavior might give us additional constraints and say maybe there's too many ligases there and we need to reduce that functionality. I don't know if that would work in practice, but these are sort of the kind of perspectives that we can take from thinking about the scaling theory. And so just to go back to the sort of idea that the last universal common ancestor might be a planetary scale phase transition, the one way to think about it, at least from this sort of physics perspective that I like to think about things, is at the genomic level, LUCA is like a surface of last scattering, because we reconstruct it genomically, but we can't really move past that to try to think about what the earlier systems were. And of course, the imprint of LUCA exists across modern Earth's surface in terms of all the genomic and metagenomic data that's distributed across the planet today. And so we can actually look sort of in terms of those LUCA components, where are they in modern systems? And it turns out that I don't have enough time to go into all the details of this, but if we look at metagenomes and we look at LUCA components and metagenomes, LUCA is almost universally present in every metagenome. So it seems to be, at least in modern systems, an eco-scale or community scale level property, which gets quite interesting in terms of thinking about what LUCA was as a population of cells, because that's certainly where it exists today. It's not in an individual species or taxa. And so I'm very interested in this idea that the scaling theory might actually help us extrapolate beyond that phase. And there's certainly some interesting features. So for like example, with the lyases, LUCA is more consistent with a metagenome than it is with some of the individual taxa. But if you look at some of the hydrolases, it's more consistent with individual taxa as far as its scaling behavior. So it seems to sit somehow between individuals and ecosystems in terms of its properties from the scaling perspective. And these are all sort of preliminary kind of considerations, but they allow us to ask new questions and start thinking about how we might be able to use this to inform new ways of thinking about construction of early life or models of life in the lab. So I'm almost out of time, and I want to make sure that we have time for questions. So I'm going to just do this last part super quick. It'll take like 2 minutes, but just to talk about symmetries, because I think it's just to show that we can also not just talk about these properties that we're looking at in terms of reactions, but also in molecular components. And so I think everyone here is probably familiar with the fact that biomolecules are often chiral and can come in a left-handed or right-handed variety, or sometimes even more, depending on how many chiral centers the molecule has. And that's very important in origins of life research, for example, because we want to know where homochirality comes from, where amino acids were chosen just to be left-handed and the sugars in RNA and DNA are right-handed. But what we've been doing is actually trying to think about labeling our biochemical network by the number of chiral centers in a molecule. So it's not looking at whether the molecule is left or right-handed, but just whether it's chiral or not, and then how many chiral centers does that molecule have, and then study the statistics of that as a window into how biochemical networks are structured in terms of the stereochemical features of molecules or their three-dimensional structure. And just as a point on this, you can also look at this sort of in a network of prebiotic chemistry. And I just wanted to say that sort of the ratio of, so this is the work on the left is from this recent paper that came out in 2020, basically trying to iterate over all the prebiotic chemical reactions known and generate a network which doesn't really represent prebiotic chemistry, but at least gives you an idea of sort of some of the chemical space that prebiotic chemistry lives in, sort of. And then what we did was just look at the number of compounds in sort of each generation that they were trying to generate prebiotic chemistry and the ratio of chiral compounds. And you see that the number of chiral compounds is increasing as you get toward more and more biomolecules being present in prebiotic chemistry. So that's obviously biology has a preference for choosing chiral molecules, but then we can actually start looking at our biochemical data. And if we look at the total number of compounds versus the number of chiral compounds, and then the inverse, the number of achiral compounds. So does a molecule have a chiral center or doesn't it have a chiral center? We also see very regular scaling behavior, and LUCA fits that model, but it's different across the different domains. And so we're working on this kind of stuff now. We're trying to understand properties of chirality in these networks and how we can think about this in terms of universal principles as well. So just to conclude, some of the design principles for synthetic life that might be derivable from the kind of work that we've been doing, and I'd certainly be really interested in talking more with people about how we can apply these to the kind of systems you work with, is this idea of scale invariant network topology. So shared components, how many shared components do you need to have a robust, say, synthetic cell ecosystem versus individuals, because it certainly doesn't necessarily need to be the ratio that life has today. It's actually a frequency distribution, but we can kind of tune that. And talk about the network properties at the individual and global scale in these systems. Constraints on the ratio of reaction functions. So I showed the data we have for the Coarse functions, at the level of the primary EC digit, but I also have a student working on second EC digits and third EC digits, and we see regular scaling behavior there. And we're working on deriving theory for why this is the case, which might provide some design principles also. And then constraints on ratios of achiral-chiral compounds, and there's some other molecular properties that come from cheminformatics we're starting to look at as well. And with that, I'm going to conclude and thank my group and all of our funders. And hopefully we still have time for questions. Sorry, I ran a little bit long.
Thank you so much, Sarah. As always, that was really inspiring. There is quite a bit of questions in the chat. Can you see that?
I can. Do you want me to just start from the top and work down, or?
Yeah, that would be great.
Okay. So I think the first one's from Denise Matthews. If the whole biosphere on a planet is a network, how would new life form fit into it? Synthetic cell that is not evolutionary connected to the rest of the biosphere. This is a great question, and I've actually thought about this in terms of this idea in astrobiology of a shadow biosphere. So there might have been an independent origin of life event that could exist today, but is kind of coexisting on Earth, and we haven't detected it yet. And I actually think my personal perspective is that this planetary constraints means that the only robust systems over evolutionary timescales are ones that have an integrated set of component reactions for the robustness arguments that I was making before. And this is one of the reasons I think about LUCA as a planetary scale phase transition. So my argument would be you have one planet, you have one kind of biochemical life form per planet, at least over geological timescales. It might be in the interim you can have multiple ones, but I don't think that those systems are going to be stable long term. Just a hypothesis, but it comes from sort of what we're seeing from the global organization of these biochemistries. And you can do sort of network experiments where you vary the overlap. And if you have less overlap, you see that the topology undergoes in the graph, the actual network structure undergoes phase transitions as you get to higher scales. So you wouldn't see networks that have the same structure at the level of ecosystems as individuals, and therefore ecosystems might not be stable in sort of a network theoretic sense, which may or may not correspond to whether they're actually stable. I mean, there's a lot of work to be done there, but I think there's some interesting hypotheses to test. Next question was from Nicholas Drachman. Is there some bias baked into the biochemical network based on what pathways and molecules biochemists have chosen to study the most? Certainly there is. Definitely bias. There can also be bias in the network construction itself. So the way you project a biochemical system onto a network can matter to the kind of structural features you see. And so that's one of the reasons that I wanted to focus on the reactions and compounds that life uses also, and demonstrate that these features were also robust in that space. But one of the reasons for doing this kind of statistical approach that I think gets at this question a little bit more directly is that a lot of times the patterns you see over whole classes of systems are sort of, they persist even if you're missing some of the details. So for example, in network theory, people will do knockout experiments where they might knock out randomly 10% of the network and show that they still have the same overall statistical features in the system. So my hope is that because we're looking at this really coarse level, that even if human bias is present in the system, or we have missing data, which we certainly do in biochemistry, because not all metagenomes, we fully know the function, or even for organisms, that these patterns would still hold true. And in fact, they can be used to predict missing features. So I didn't talk about this, but with the enzyme scaling, for example, if you take a metagenome and you have a certain ratio of enzyme functions in that system, you can kind of predict what the missing functional diversity is based on the scaling constraints. So there are some reasons there that we think that this might help with some of those issues about, like, could we use it to actually say how much bias or how much missing data there was there? And especially if we have theory to predict it, then we could actually say under what circumstances we'd be able to do that. That's a good question. So John Bartel's question is, what if you engineer biochemical network from scratch? It's not natural complex network, but it's engineered. Would it look like life or non-life? I think it depends on what you engineer into the system. This is a really good question. I'm super interested in trying to think about this. So I think one of the ways of interpreting the scaling behaviors that I was showing is that life has evolved toward this optimal, right? So there might be some physical constraints on the structure of biochemical networks. What I mean by that is, so it's easy to see sort of with redox reactions, right? Redox reactions scale super linearly with network size, or with the number of total enzymes in the system. And intuitively, that seems like maybe that should be the case, because redox is associated with energy harnessing and energy use. And so maybe it's just that larger networks have more energy demands than smaller networks. And it's not a linear scaling, it's actually super linear scaling. So you have much more energy demand at sort of larger network sizes. And I think what we could do with that is, if you engineer a biochemical network and it's not evolved to the optimal, it might have it might have different properties than sort of these optimal network configurations, and maybe that plays into the design. But what I would like to do is actually, if like I'm saying, like we can understand why these properties are there, we can talk about sort of the statistical parameters that define what a living network is. And then if you have an arbitrary engineered or prebiotic chemical network, you can say, like assign a statistical confidence, it's actually part of that living ensemble or not. And it tells us sort of a little bit, it should give a measure of how alive it is, not just whether it's alive or not. And so I'd be really interested in that. I think we're right at the hour, Kate. There's a couple more questions. I don't know if you want me to go through them or you want to wrap up.
I think if you can go through them, if someone needs to go, they can log off. But I'm actually curious to hear your answers to some of them.
Okay, so Aran Agman asks, is it possible to identify universal properties of autonomous biochemical networks which continually maintain themselves? I think finding such properties might be a good way to delineate cellular or organismic scales from within chemical space. Yeah, no, I definitely think that's possible. So the same kind of biochemical networks that we're using, people have, for example, looked at whether there's autocatalytic sets within those networks, not as large an ensemble of networks as we have, but like maybe an E. coli or specific examples of things that might be relevant to origins of life chemistry. And so if you think of like autocatalytic feedback as sort of the self-sustaining thing, you could, apply the kind of statistics that we do basically to that principle. Although I actually have some trepidations about making the argument that because you see a cycle, like an autocatalytic cycle in a graph, that system's actually stable as an autocatalytic system in the lab or in real life, for a variety of reasons that a lot of the work on autocatalytic self-sustaining systems has been done in graph theory and not in dynamical systems, and they're often not stable in the way that you would think that they were. So I think that there's a lot of work to be done in that regard. But I do think that some of the features that we see as far as the component set of universally shared reactions and things are related to the self-sustainability of living networks, and some of those features we should be able to understand from this kind of approach. But actually that's one of the reasons that I wanted to study chirality, because I think chirality constrains reaction space in such a way that you're more likely to get closed loops and things. And so it's actually kind of necessary for reproduction, which is kind of related to the way people talk about it in homochirality, but in a slightly different perspective. And just one way to think about this, like if you have a reaction that has chiral, like chiral centers, if you have one of the sort of enantiomers, the reaction space is constrained to go this way. And if you have the other, it's constrained to go that way. And so you can actually imagine that these are sort of constraints and sort of the funneling of like how things flow along pathways. And then that can allow you to maybe get some features of what we would call more autonomous systems. So I think there's a lot of rich questions to ask there, basically, and a lot of different directions to go. And then Emmanuel Bernard asks, can the universality model help predict minimal synthetic metabolism? I hope so. I think what I'd like to be able to do is just try to, like, for those scaling laws, try to see sort of like what would the sort of systems at the scale of, minimal metabolisms actually have to have for their component properties, and could we build features of those systems. So one of the projects that we're working on now is at least focusing on LUCA, and the way I think about constructing the last universal common ancestor is to build an ensemble of multi-scale models that have, like across, taxa and ecosystems at the same time, to constrain sort of statistical structure of what a LUCA model might look like. And I think you could certainly try to build minimal systems based on that, or even just do it in the metabolic reconstruction that way. I think I went through all the questions. I don't know if there's any other ones anyone has.
Thank you so much.
Sure. Thank you.
And I really think we need analysis like that, someone to just study the metabolism and tells us this is exactly what we need to build to have an actual self-sustaining synthetic cell.
Hopefully we'll get there. Let's have some like real numbers to give you one of these days. But we're getting closer. Yeah.
Definitely. All right. Thank you so much, Sara. I really appreciate.
Yeah, thank you. Thanks,
and
everyone.
Thanks everyone, and bye.
Bye.